Hello everyone, welcome back to my channel. This is Sana's Biochemistry and I am Sana Parveen. In this video, we are going to talk about coronavirus and COVID-19. We are just going to talk about the biochemistry of the virus so that we have got better insights about what the virus actually is and how is the virus entering the different types of cells or entering the cells in our body. So let us just see about that. Let us move into the video. Okay, so coronavirus, if we talk about that, it is coronaviruses are a group of related viruses. That means coronavirus is a family of virus. Okay, so it has got a family of virus in which that causes diseases in mammals and birds. So coronavirus is going to cause diseases in mammals as well as birds. In humans, coronaviruses cause respiratory tract infections that can range from mild to lethal. So what which system of our body is affected by the virus? It is the respiratory tract or respiratory system is affected, and the infections can be mild or lethal as well. Mild illness includes some cases of the common cold, while more other lethal varieties can cause SARS, MERS, and COVID 19. Okay, if we look at the full form of SARS, it is severe acute respiratory syndrome. MERS, it is Middle East respiratory syndrome, and COVID 19 because it is called as coronavirus disease 2019. Why do we call it COVID 19? Because it has got a full form of it, which is coronavirus disease, and 19 is for 2019. Okay, 2019. So this is the reason why we call it to be COVID-19. It is just a short form of it. Let us see ahead. Why is the name corona? Okay, there are different infections that we have seen, like swine flu, bird flu, etc. This is specially called as corona because it has got a reason. Let us just look at the reason why is it called as corona. Before we move on to that, we just need to know that coronavirus is a group or family of viruses in which we have got various different types of viruses. It can be, causing SARS, it can be causing SARS, it can be causing MERS, okay, or it can be causing COVID-19. So it's a family, and the family has got different members in it. Now we are going to look at why the name corona. They have characteristic club like spikes. The virus is going to have characteristic club like spikes. The spikes which are present are like red in color, that projects from their surface, as you can see, which in electron micrograph create an image. If you see it under an electron microscope, it is going to create an image like a solar corona, like an image like this. A corona, which in Latin means crown, is an aura of plasma. So what is corona exactly? The meaning, the literal meaning is we've taken from Latin, and the word is crown in Latin, is an aura of plasma that surrounds the sun and other stars. The sun's corona extends millions of kilometers into outer space and is most easily seen during a solar eclipse. So when solar eclipse happen, it is going to look like this, and similar kind of structure is present in this virus. So the name is coming from there, coronavirus. Okay, let us move ahead now. Coronaviruses are enveloped viruses. Okay, here comes the difficulty. It is an enveloped virus. That means the virus is going to be there. Okay, the virus is there, and it has got an envelope on top of it. Okay, so enveloped viruses are difficult to handle. So it is an enveloped virus. Most viruses like HIV are also enveloped viruses, or they have got a viral envelope. So this is nothing but the viral envelope. So HIV and many other animal viruses, they have got a viral envelope as their outer layer. This is present as an outer layer, and the envelope is giving it, something called as extra protection. Okay, it is highly protected. The virus is sitting nicely inside, and the envelope is surrounding it. The envelopes are typically derived from portions of the host cell membrane. So where is it deriving the envelope from? When it is getting it from the host cell. For example, this is the host cell, and from the host cell it derives its envelope. And as the host cell, the cell membrane, we everyone knows it is made up of phospholipids. Okay, so as it is derived from the host cell, we will conclude, or we can conclude, that envelope of the virus is having or is made up of phospholipids and some proteins, but instead includes some glycoprotein as well. So as you can look in the structure here, let us just look at the structure here. In the structure you can see this yellow color is the envelope, which is giving an extra protection to the virus. For example, there's a boy, okay, and the boy is standing there, and it started to rain. Okay, it started to rain from the clouds. The boy had a raincoat, so he wore the raincoat. Now he's protected. Now the rain cannot have an effect on the boy. The similar thing is that the envelope is causing the problem. The envelope is protecting the virus, okay, and it is not letting the virus be killed by something. So this is the envelope, okay, made up of what? As you can see here, it is made up of phospholipid and is derived from the host cell membrane. It has got antigens present on it. It has got the envelope. It has got the capsid. Capsid is formed from globular protein, and there is this genetic material which can be RNA or DNA. If we talk about coronavirus, we have got the genetic material to be RNA, ribonucleic acid. Let us move on ahead now. Let us see what do we have next. Okay, they may help viruses avoid the host immune system. As I said ki, these viruses, they have got extra protection around them, okay, a protective layer around them, the envelope around them, which is helping them avoid the host immune system. So if this is the host is there and the virus is now entering into the host body, the host immune system, the immune system which is involved in the protection of our body, that is not having an impact on the virus, because the virus is coming inside with a protection on it. So they help, the envelope helps the virus avoid the host immune system. Glycoproteins on the surface of the envelope serve to identify and bind to receptor site on the host cell membrane. Okay, now what is it doing? The glycoproteins on the surface of the envelope. So if this is the envelope and there is this virus inside the envelope, as we saw, there's something present on it. This is nothing but it is glycoprotein. Glycoprotein means what? Oligosaccharide chain, or it is also called as glycan or carbohydrate with covalently attached amino acid side chain. So it is a combination of carbs and proteins. So this glycoprotein which is present on the envelope serves to identify and bind to the receptor site of the host membrane, host cell membrane. So if this is the host cell membrane, the virus is going to go and identify what the receptor site, where should I go and fix. It goes and fixes, and then everything, the havoc happens. The viral envelope then fuses with the host membrane. The viral envelope, this envelope is now going to go and fuse with the cell membrane. Now the envelope is fused with the cell membrane, allowing the capsid with the viral genome to enter and infect the host. So now, for example, this is the capsid. Capsid enters with the genetic material, the RNA, into it, and the viral envelope is going and fusing with the host cell membrane. So we that is the reason why we need to be extra careful when we're talking about enveloped viruses, because these viruses are difficult to handle. But we have got certain advantages. What are the advantages here? The lipid bilayer, which is the envelope thing, envelope of these viruses is entirely is like relatively sensitive to desiccation, heat, detergents. Desiccation is like removal of water, heat, okay, because of which we're saying that high temperature and everything, and the detergents. Therefore these viruses are easier to sterilize than non-enveloped viruses, because now what is going to happen, this is the virus here, okay, the deadly virus, and this virus has got a sensitive lipid bilayer around it. Okay, this lipid bilayer can be destroyed by the three agents. So the layer can be destroyed, and now as the virus is exposed, what we can do is we can take actions and can the virus can be taken care of. Okay, now what do we have? These viruses are easier to sterilize than non-enveloped virus, And what are the advantages that we have limited survival outside the host cell. It is not going to survive for much of time, because there's a controversy about viruses, okay, which I have made already a video. I'll be providing you the link in the description below, so you can go and watch that video as well. So limited survival outside the host environment and typically transfer directly from host to host. As we said that coronavirus is an infectious disease, it is like spreading. Coronavirus, that now we come to the more important discussion. Coronavirus are positive sense single stranded RNA genome. So if this is the virus, the RNA which is going to be present in it, first of all it is single stranded, okay, single stranded RNA ssRNA, and then it is having a positive sense. Now what is the meaning of positive sense? Single stranded RNA virus are classified as positive and negative depending upon the sense or polarity of the RNA. Okay, so positive sense is running from, where is it running from? It is running from 5 prime polarity to 3 prime polarity. Okay, so that is the reason why we say positive sense. Negative sense is going to be different, 3 prime to 5 prime. This is something about coronavirus, which is their positive sense and single stranded RNA genome. Genome means the genetic material, and this is the positive sense, 5 prime to 3 prime. Let us move on ahead now. Okay, positive sense viral RNA. There's one more important thing about the virus. Positive sense viral RNA signifies that a particular viral RNA sequence may be directly translated into viral protein. Positive sense means what? The RNA is ready to be translated into viral protein. Those needed for viral replication, the viral proteins which are needed for viral replication to make numerous copies of the viruses, because one virus is going to make thousands of its own copies. Okay, it is like xeroxing itself. Okay, so now the viral RNA sequence, it is like so ready and is not needing anything, and just translation will happen, thousands of viruses will be produced, and then the disease can spread like so fast. In positive sense RNA viruses, the viral RNA genome can be considered viral mRNA. Okay, the mRNA, what is the meaning of mRNA? Is the messenger RNA. It has got the message, okay, how to synthesize the virus. It is like so ready. Okay, it is like instant 2-minute thing, not even 2-minute thing. Okay, it is like so instant that it needs only translation, and the virus is going to be born or like pop out and not much time, and can be immediately translated by the host cell. Because of this, these viruses do not need to have an RNA polymerase packed into the virion. The virus is not needing anything. As I said, RNA polymerase is not needed by the virus. Okay, it is other viruses need that. So there is no RNA polymerase present in the virion to have an effect on it. Before mRNA can be processed, it must first be transcribed by RNA polymerase. So this step is like completely skipped, and the RNA polymerase is like so sad that the corona is not making him sit in his inside itself. Okay, so next, coronavirus disease, the full form of COVID-19 is, for the information, coronavirus disease 2019 is an infectious disease caused by whom? Caused by SARS-CoV-2, that is severe acute respiratory syndrome, coronavirus 2. The disease was first identified in December 2019 in in Wuhan and China. Okay, resulting in the ongoing 2019-2020 coronavirus pandemic, okay, throughout the world. Let us just look at the structure of the coronavirus now. Okay, so we have got the envelope. As I said, the envelope is present. The envelope has got protein inside of it. The yellow color thing is the protein. It has got the membrane protein, the U-shaped thing, spike protein, okay, like a crown, which is giving the appearance like a crown. Spike protein, which is going and binding to whom? To the host cell, and then causing all the havoc. Nucleoplasmid protein with enclosed DNA. So this is the nucleoplasmid protein, and it has got DNA enclosed inside of it. The DNA is not exposed out of it. Okay, so in this green color thing, let me just draw the RNA, RNA. Okay, inside. So it is having nucleocapsid protein enclosing RNA, and there is this lipid membrane which is forming the envelope and giving protection to the virus. Okay, let us move on ahead now with this coronavirus entry point. This is like so interesting. Coronavirus entry point, the lungs are the organs most affected by COVID-19 because the virus accesses host cells via the enzyme called as ACE2. So who's the virus is taking the help of whom? It is ACE2. What is the full form of ACE2? Angiotensin converting enzyme 2, which is most abundantly in the type 2 alveolar cells of the lungs. So if I draw the lung, I've drawn the lungs, and in that lungs we've got the cells in the lungs. Okay, so cells in the lungs. On the cell you have got something sitting. Okay, okay. Actually, it is present in the cell membrane. Let me just make the cell membrane kind of thick. This is the cell membrane in which there is ACE2 sitting. The cell membrane has got ACE2. It has made place for ACE2 to sit inside of it. So this is ACE2. ACE2, who's helping H2? Okay, now comes the virus. Now comes coronavirus. Coronavirus is going to take the help of H2 and then enter into the cell. Now let us see how. Okay, so we have got the H2, the helper, okay, or the entry point of. So H2 is like so sad to help coronavirus. Okay, not that happy. H2 is there, and then comes the deadly coronavirus. Okay, so this is virus and this is H2. H2 is present where? It is present in the cell membrane. Okay, coronavirus is going to take the help of H2 and then inject its genome inside. Okay, let us move ahead. Angiotensin converting enzyme 2 is a zinc containing metalloenzyme. It's an enzyme located on the surface of endothelial and other cells as a transmembrane protein. What do you mean by transmembrane protein? Okay, so if this is the cell, it has got the cell membrane. I have drawn cell membrane like so thick. So let me just, make it more clear. This is the cell membrane of the cell. Now where is ACE2? ACE2 is uh okay, ACE2 is here. The red colored thing is ACE2 present in here. Okay, and this is now going to, when the corona comes, okay, this is now going to take the help of ACE2, and then the genetic material is going to be, the mRNA is going to be injected, like already making new RNA, making new viruses, and then viruses are going to be spreading out all the time. ACE2 serves as the major entry point into the cell for some coronaviruses. The virus uses a special surface glycoprotein called as the spike to connect to ACE2 and enter the host cell. So this coronavirus is having the red color thing which I was telling you about. Okay, just let me draw it in black. Okay, I hope everyone is able to see that. That black color tiny thing is, the black color tiny thing is what is the virus comes and shows its spike to the ACE2, and then it can enter into the host cell. The density of ACE2, the density of ACE2, now let us see. The density of ACE2 in each tissue correlates with the severity of the disease in that tissue, and some have suggested that, okay, that means if the cell is there and ACE2 are present like more in number, then what is going to happen? It will help more coronavirus to enter into the host cell, like everywhere the host cell and the coronavirus is having a spike on it. The spike is going to connect with the ACE2 and can enter into the cell. So more number of ACE2s, more chances of infection, and to a very severe degree. The density of ACE2 in each tissue correlates with the severity of the disease in that tissue, and some have suggested that decreasing the ACE2 activity might be protective. Okay, so just decrease the activity of ACE2. Don't let the ACE2 work. Now, how can we not let the ACE2 work? By increasing ACE2 using and by increasing H2 using angiotensin 2 receptor blocker medication. So give the medication in which the medications will come, and the medication will come and block the receptor. Okay, the receptors or angiotensin 2 receptors are actually G protein coupled receptor, which are also called as GPCRs. This GPCRs or G protein coupled receptors are family of receptors that detect molecule outside the cell and activate what the internal signal transduction. Okay, let me just explain you this one. There is the cell, okay, and then you have got the H2. The H2 has a receptor. The receptor is present in here. Okay, let me just take it. Okay, then what is going to happen, this angiotensin is going to help the virus. The virus is present in here. The virus is going to show what? The virus is going to show the spike is going to connect to the H2, the red color thing. The ACE2 is having what? A receptor, the G protein coupled receptors. Okay, so this yellow color thing is what? GPCR. So once the signal comes, okay, the virus is there. Once the signal comes, this is going to spread the signal into the cell, and then havoc happens. So if we take care of this part, something can be done, something or other can be done. As the alveolar disease progresses, respiratory failure might develop and death may also occur. Okay, as we can see, which is happening in the world, all around the world. Okay, so how is the diagnosis done? The diagnosis is done by the technique called as real okay, the technique called as real time reverse transcription polymerase chain reaction. Okay, so this is the technique by which we are doing the testing, RTR PCR from the nasopharyngeal swab. Okay, so this is the diagnosis part of it. And then thank you for watching the video, and if you like the content, hit the like button, subscribe to my channel, hit the bell icon as well, share it with your friends so that the people get the correct or useful knowledge. And then stay at home, stay safe, plank the curve. Thank you once again, and bye bye. See you in the next video.